Welcome to Module 3, the cells and cellular activities of the immune system to include granulocytes and mononuclear cells. When we have cells in the immune system, you guys are learning a lot about the cells in hematology if you're in that course right now. And you're learning about what they do and what they mean, but what you don't realize sometimes is there's a lot of talk going on between these cells. Okay, and here's an example of some of the things that we might see. We have a T cell, CD4 cell here in the middle. He's sending messages to other cells, dendritic cells, to kill bacteria. Bacteria are, doing certain things. Neutrophils are only phagocytizing bacteria. They're also creating inflammatory types of products. We've got cytokines to try and call in more cells. While I don't expect you to memorize this, just know that there's a lot of talking going on between the different cells to maybe call out for more help, per se. So some of the things we're going to talk about are some of those cells and exactly how they work in the immune process. All right, so first let's look at how these cells are developed. And by the way, if you're in Hematology 2 right now, you're going to see a lot of overlap in this chapter. Things are going to start looking really familiar, which is kind of fun. So the first thing that happens, the first 2 to 8 weeks of life, we have erythroblasts formed in the islets of the yolk sac. 2 to 5 months of gestation, the liver and the spleen are the major sites of hematopoiesis, and granular leukocytes also appear. In the 4th month of gestation, bone marrow begins to produce blood cells, and the 5th month, bone marrow is the ultimate rule of hematopoiesis. You must know that it starts in the yolk sac, then it goes to the spleen and the liver, and then to the bone marrow. This you do need to know for sure. So what are considered granulocytic cells? The cells that have granules. Hopefully you're familiar with these from your hematology course, but we're going to review. Neutrophils provide the host defense against bacterial and fungal infections. Its primary function is phagocytosis, which is the eating of bacteria, kind of like Pac-Man. Eosinophils are homeostatic regulators of inflammation. Their primary function is to suppress an inflammatory response. They also play a role in killing parasites. Basophils play a role in the acute systematic hypersensitivity reactions. When these puppies degranulate, when the antigen binds to the IgG, it releases heparin and histamine. That is what makes you itchy and sneezy. That's why during the flu season you take an antihistamine to try and suppress that response, because it makes you uncomfortable. We talked about phagocytosis, which is the actual eating of bacteria by the cell. Okay, there is a process, and I have on the bottom CAEDD, can an elephant dig dirt? That is how you can memorize this. The first one is chemotaxis. That is the process that promotes the attraction of the phagocytic cell towards the foreign particle by cytokines. Okay, so chemotaxis, it's like taxi, is movement. You take a taxi, taxis sounds like taxi. It then adheres, okay, to what it's going to be destroying. And before it got there, opsonization had occurred. It was the coating of organisms by something called C3 molecules that speeds up the phagocytosis. It's what makes it super tasty. I think of it as the body putting salt and pepper on the organism to make it extra tasty. That's what opsonization is. It adheres to it. It then begins to engulf it, like Pac-Man. It digests it, which is where the dig comes in, and then it destroys it. So can an elephant dig dirt is the process of phagocytosis. Here's what it actually looks like. So first you have a bacteria. Through chemotaxis, the neutrophil is attracted to the bacteria. It then adheres. It becomes engulfed. It then becomes a phagosome inside the cell. It all fuses together, and then it's digested and destructed. It's important to make note that this really should only happen with foreign organisms. We do have normal flora in our body, which are microbes that do not cause disease. Here's another picture. We have the live bacteria. It is being engulfed. Inside, there's soluble serine proteases, etc., that kill the bacteria. Inside, a phagolysosome is also formed. That is a structure within a phagocytic cell by the fusion of the phagosome with a lysosome. This helps with the destruction. Our next type of cells are monocytes and macrophages. The role of the macrophage in cellular immunity is to ingest foreign particles and infectious microorganisms by phagocytosis. He's also a great antigen presenting cell. So not only does he eat garbage, he goes home to mom and shows him the garbage that he made. So he's able to take the antigens from the things that he's eaten and show it to the T cells and etc. and say, hey, look at this. Remember this? Make sure you help us destroy it next time you see it. And here's just another example of what those might look like in tissue. Macrophages are found all over. They're named different things depending on where they are. In your nervous system tissue, they're called microglial cells. In the lymph nodes, they're macrophages. In the bone, they're osteoclasts. In the spleen, they're macrophages. They're pulmonary alveolar macrophages in the lungs. We call them Kupffer cells in the liver. Glomerular mesenglial cells in the kidney. And tissue macrophages or histiocytes in your tissues. So they have, while it is a macrophage, they kind of have different roles in different areas. The functions of the monocytes and macrophages in host defense, make sure you memorize this list. I'm kidding. It's just an example of some of the things that happen. If you have increased activity in activated macrophages, some of these things might be taking place. Antigen presentation, chemotaxis, etc. Increased constituents in activated macrophages, some other things that we find. It's just a big list of things that happen, different chemicals that are taking place to help with the destruction. So do not memorize this. I was totally kidding. All right, so with monophages and monocytes and macrophages, what do you really need to know? Well, antigen presentation. That is the induction of the immune response. So remember I said those macrophages eat the garbage and then they bring it home to mom and show them what they made? That is what antigen presentation is. It can take the antigens from what they've destroyed and present it to the rest of the immune system. The secretion of biologically active molecules, which is called opsonization. Remember, that's what I called taking the bacteria and putting salt, pepper on it to make it extra tasty. Essentially, that's what opsonization does. Here's some interesting facts for you. Have you ever heard of people taking corticosteroids or getting a steroid injection in some part of their body that's inflamed? Like an athlete getting it in their knee or their back. Some people get steroid injections in their back. Some people get a rash, a big puffy, itchy rash, and you put a steroid cream on it, such as cortisone. So what that is, it's a steroid therapy that can cause a defect in phagocytic killing in monocyte macrophages, so it stops the inflammation from happening. Diabetes and burns can be depressed, can cause depressed migration of monocyte macrophages, so that can cause an issue. If you think of some diabetic patients that have poor circulation in their feet, they get infection. Not only does the circulation cause a problem, but the migration of monocytes and macrophages being depressed can cause issues with the healing of that area as well. Intracellular infections can cause defective release of macrophage activating factors. So we have to be careful. Here's an example of acute inflammation, or let's say an infection in your skin. So this up here is your bloodstream, a blood vessel, okay? We have a little neutrophil rolling along, and it comes across this Cellular X coming out of the side of your vessel wall. This L-selectin actually sheds, and integrin comes into your Cellular X, creating E-selectin. This, and you don't have to memorize the Cellular X, the L-selectin, or the E-selectin, but just be aware that as it rolls along, something causes it to adhere to the side of the vessel wall. What's really important is to know the term for the movement of the white cell through the vessel wall, that is diapedesis. Through diapedesis, it makes it through the wall. And things like C3A, C5A, you'll know what those are in the next couple weeks. Chemokines, histamine, prostaglandins, leukotrienes, etc. help this process along. So now we're inside the tissue. Now that it's in the tissue, it's able to phagocytize and destroy C3b coated bacteria, which are what opsonization is. Opsonization is the C3b coated coating. Okay? So that is the process of diapedesis, the white cell moving through the blood vessel into the tissue. A term you need to be familiar with is sepsis. Sepsis is an infection or inflammation-induced syndrome that overwhelms the whole body. It's defined as the presence of two or more of the following. And these counteract each other, I apologize, but we do see both of these. Fever or hypothermia, leukocytosis or leukopenia, tachycardia or tachypnea, which is low cardiac heart rate, and having supernormal minute ventilation. So how you breathe would be affected as well. We're going to look at some disorders of neutrophils, and these also with the monocytes, and these are the ones you've probably heard of in your hematology course. Neutrophils as mediators of non-infectious inflammatory diseases. So some of the disorders you'll see can either be quantitative or qualitative. Quantitative means there's a problem with the number of them. You could have neutropenia, which means you don't have enough. A qualitative defect would be something like the ability of a leukocyte to move. So there's something wrong with the quality of the leukocyte. So these are the disorders I want you to be familiar with. The first one is Shediac-Higashi. With this one, we see delayed killing due to giant granules with impaired chemotaxis. The next one, chronic granulomatous disease, or called CGD. You can refer to that in the bottom right-hand corner as well. This one has issues with the cascade of events required for hydrogen peroxide production or failure to exhibit increased anaerobic metabolism during phagocytosis. All right, so how do I memorize a couple of these? I don't have a good thing for all of them, but CGD, I think of granulomatous. Have you ever wiped out on gravel and you stand up? I know my kids have. They stand up and there's all kinds of rocks embedded in their hands, like little rocks. Well, I always think of going home and my mom pouring hydrogen peroxide on it. So I've got this granulomatous stuff that I pour hydrogen peroxide on. So whenever I see a disease with hydrogen peroxide production failure, I think of the chronic granulomatous disease. Systemic lupus erythematosus, SLE, that one can cause impaired phagocytosis. We have a whole chapter on that at the end of the quarter. Myeloperoxidase deficiency, this one is a defect in the bactericidal ability of neutrophils. There is no hydrogen peroxide there either. And leukocyte adhesion deficiency, or LAD, is a deficiency of integrin. Remember that little molecule? It doesn't allow the neutrophils to adhere to that vessel wall. Couple others, monocyte-macrophage disorders you need to be familiar with. First one's Gaucher's disease. That one represents a deficiency of beta-glucocerebrosidase enzyme. With that, we'll have issues with destroying foreign organisms as well. When I see Gaucher's, I put that with glucocerebrosidase. They both start with a G. Niemann-Pick disease represents a deficiency of sphingomyelinase, which is an enzyme. It's an abnormal metabolism being present within the cell. They look very foamy. Okay, so this cell down here, I'm trying to point to it here, it's a very foamy looking cell. And when I think of Niemann-Pick and I think of sphingo, to me it sounds like finger. So you pick your nose with your sphinger. Okay, so that's how you memorize that one. Also, it's very foamy looking, and my kids' snot when they blow their nose is always foamy. So again, I apologize for the horrible alliteration of that, but if it helps you memorize it, it helps memorize it. One of the other ones, Wiskott-Aldrich, that one is a defect in monocyte toxicity, so they get infections frequently as well. This one I mentioned already, the leukocyte adhesion deficiency. It's characterized by the impaired leukocyte adhesion. Remember that integrin molecule becomes the problem there. Often fatal bacterial and fungal infections with them. The last one, lazy leukocyte syndrome, is a defect leukocyte locomotion. So the cells are not moving as well as they should. All right, well that concludes Module 3. Nice, short and simple. Next week we'll be moving on to the lymphocytes, which are a little bit more complicated. But have a great week.